Hello and welcome everyone to the first Zangwill Club of the year, not the academic year, the actual year of COVID, second year of COVID, 2021. And we would normally have Hakwan in the premises of Cambridge for this, but since the pandemics, started last year early on, if you didn't notice, then you've been living in another world. We are doing this online, and it's been going really well for the discussion and for the talks so far, except those times that we had some technical difficulties, but hopefully today will be okay. The exits are at the end of the room. In 2004, when I went to my first consciousness meeting, I found a young guy who was very serious about science. And he introduced himself as Hakwan. And then we went for drinks. And I thought, this guy says he's very serious, but he's also quite fun. So I don't know why he says he's so serious. And it ended up being that he was both. He was very serious about the science, but it's also a guy who can relax and have fun and actually likes to have fun. You can move forward there, from 2004 until 2011, then this same guy is taking you to funny places in Japan, to meet people who own bars around poets, and they have special type of sake and all of that. So if you want to read about, Hakwan's academic accomplishments, you can definitely do it. And these are all over the web, and in his website, his webpage, in a Scholar Google. He's an accomplished scientist. He's someone who's an activist of science, I would say. And he has strong opinions about, theoretical aspect and experimental aspect, but at the same time, he tries to listen to the other views. He might mention something in the talk today, and he will love if you actually attack him scientifically at the end, because he really likes a good discussion. So hold your horses for a good discussion at the end. If something is not clear, please, say it in the chat, and maybe we need to interrupt. Most likely you won't, because he tends to be very clear of what he explains. And with this, I would like to welcome Hakwan. Welcome to the Zangwills.
Thank you. So thank you, Tris. Although you set the expectations, way too high for me, but I also thank you for not mentioning the more embarrassing episodes of our adventures. So I'm very grateful and glad to be here, seeing a lot of old friends and new friends. So today I'll talk about actually something it's a bit of a cheeky title, because it's some work that I only started to dabble in a little bit. I'm not talking about the kind of coma or vegetative state, patient-related kind of work. I'm talking about different kind of way you can take mostly the conscious perception literature into a slightly clinical direction. But it's quite new, so some of those will be empty promises. Hopefully may not be empty, but thus far promises so far. But I'll talk about some old work too. So, okay. So I'm interested in consciousness, and I'd like to sometimes explain to students and other people what does it mean by saying, well, I'm interested in what's so special about the human brain that make us not like computers these days, maybe not even like computers in principle. Maybe there's something that is really special, because computers aren't meant to feel anything. I mean, we don't think the current computers do. In this paper, we kind of speculated a little bit. Maybe you can build computers to make them conscious, but it's really pretty speculative. I like to think at least current computers and current robots do not really have subjective experience. They don't feel pain, like the painfulness of pain. They might be able to sense a signal and avoid it, and etc. But they don't have this subjective experience. So I think this is maybe a uniquely human thing. And now some of you might think this is so foolish, because I don't own a pet, so maybe that's why I'm so foolish. But other people who own pets would tell me, of course, pets are conscious. So on the internet, you have videos like this. They're clearly showing that animals can experience emotions. It's one of my favorite videos. I watch it again just for my own entertainment. So this dog looked really mortified, right? I mean, it clearly experience emotions. And if you interact with animals more, you would probably be more convinced by even the video like this. They clearly have some sort of subjective experience. But then you can also think about a plant like this, a plant called Mimosa pudica, and sometimes it's called a shy plant. So when you touch it, it kind of shies away. Again, it looks like it has subjective experience too, right? By the same kind of logic, and that's why it's called a shy plant. But I think very few of us think that the plant actually has subjective experience. There are some people who think so, but you may not want to talk to them for too long. The issue, of course, is we anthropomorphize and project our psychology to whatever we see. So even just a drop of water in a boiling pot of oil would look like it's really struggling and moving very vigorously. And of course, we can't just, the moral of this is you can't just project your own psychology and do this kind of guesswork. So this is an entirely not novel idea, but I think decades ago, people have already started thinking, well, maybe one way to go is to go to the human case where we know when people are conscious, when they're not, because I mean, maybe know is a loaded word, but we can be fairly sure, or more sure than in the animals and robots and plant cases. And then compare cases when they're conscious and not conscious and look for the neural correlates. And then from there, we understand the mechanism. And then from there, we can then see how this mechanism generalizes to other animals. That is, whether other animals may have similar neural correlates or mechanism or not. So this is a fairly old study. I'm skipping a lot of details just to give you a flavor of kind of where I come from. So this is a patient's brain. This is patient GY, the famous blindsight patient who has the left primary visual cortex lesion. And so you can present stimulus to his right visual field, and he was said he has no clue what was presented. He basically, according to his self-report, he lacks subjective experience in the blind field. And then in the other visual field, which served as a very good control, because for that part of his brain is completely intact, and you can have a normal field. So he's a normal sighted field, so you will see the stimuli. And you can present simple shapes. So here are basically a pair of gradings. So it's like vertical, horizontal, vertical, but the details don't matter. Basically present some simple shapes and then ask him to press keys to indicate two alternative false choice, which was the stimulus. So you can actually present a very strong stimulus to the blind field so that he will be able to guess it at 75% correct, so well above chance. So that's the amazing finding of blindsight, that is, despite his denied subjective awareness, he actually, when forced to press keys, he actually can do it well above chance. But what is good about this patient from an experimentalist point of view is you can also present a weaker stimulus to the normal field, so that it won't be at ceiling level, because you can titrate it exactly down to 75% as well by making the stimulus weaker. So you match task performance in these two conditions. And then after each trial, you can ask the subject whether you actually saw the target. And you can see that in the normal case, I saw it about half of the time, which kind of makes sense because it's a near threshold stimuli. But using the same criterion and say, like, basically, if you get a hint that you saw something, what it is, then you can rate. And in blind field, basically just very few trials that he would say some. He would say that he saw something, and mostly it's from light scatter from the other field. If you actually probe him a bit more, but you can discard those trials anyway. And then you have basically a task performance match condition that you can compare these two conditions using fMRI. And then what we found was that there was actually pretty widespread activity in the frontal and parietal areas. And that you might think is trivial because a lot of people say that the frontal parietal areas are important for consciousness. But I'm gonna say when I first saw this, I was quite surprised, because I thought a lot of those frontal parietal activations reported by people like Stan DeAnne, I thought that they were all confounds. I thought they were all just reflecting superior perception versus very little perception. So think about the normal experiment. Let's say you do visual masking. When you unmask the stimulus, you saw the stimulus, and of course you can perform very well. And when you mask the stimulus, your performance might be a chance. And I thought that, yeah, so of course consciousness differ between the two of them, subjective experience differ, but also in one case you can do a lot of information processing. In the other case, you can do virtually nothing. So I thought that all these frontal parietal activation would just reflect that. But here you have it completely controlled, and yet you find these strong differences that seems to be more specifically driven just by the difference in having subjective experience or not. So it's not just driven by a map. This is not just perception or information processing. Seems to be more subjective. And of course, you can do this. This is just one patient. You can do this in with subjects from the normal or from the general population too. These are undergrads subjects, so they're quite specific. You may not think that they're neurotypical, but they reflect a general population. And you can use other psychophysical tricks such as metacontrast masking to mimic some conceptually similar comparisons. So these are not brain lesion patients, but you can actually use metacontrast masking and create a pair of conditions under which you have the same task performance. Again, it's a simple shape discrimination, a square or rhombus, but the details don't matter. But what is interesting here is by varying some subtle timing of the mask, you can actually create a matched performance condition where the subjective rating of awareness differ. Of course, it's not so dramatic as in blindsight because these are actually healthy volunteers, but you can create a consistent difference and then again put them in the scanner and compare, and then again you would find these fairly consistent differences that are specific to just a difference in subjective experience. And here in the dorsolateral prefrontal cortex, I'm not denying that you may find parietal difference too. It might be a statistical sensitivity issue. In fact, I would not even deny that you might even find visual cortical difference there. So I think there's sometimes a misunderstanding, as if the people who believe that the NCC is part of the prefrontal cortex is part of the NCC. We usually don't deny that the other areas are important. It's quite the other way around. Is that some of our opponents would say that every other area is important, but the prefrontal cortex is decidedly not important. We just want to reject that kind of rather strong claim. So here, at least in the absence of observing some other activity, we found that this still survived this rather tightly controlled comparison. So that's quite interesting. So that got me thinking maybe the prefrontal cortex was part of the NCC. But of course, some people think that it doesn't make any sense because when you lesion the prefrontal cortex, they would claim that it doesn't affect your subjective awareness at all. Turns out it's very controversial because if you just lesion unilaterally the prefrontal cortex, in fact, it doesn't abolish much of other prefrontal functions anyway. Because I've also met patients with basically the entire basically one hemisphere of the prefrontal cortex gone, and he says he enjoyed playing chess. So a lot of these working memory, whatever other function, if you just do unilateral lesions, we know that it often does not do a lot of damage. And if you find bilateral complete lesions, and then there are some reports that those patients are still fine, but I think those claims are factually untrue. If you go into the details, most of those patients do not really have complete lesions. And if you really find complete lesion patient, I can actually show you a video. I can actually link to it later. That actually those patients are basically very difficult to test. They are virtually unresponsive to external stimuli. So it's difficult to test it, and we thought maybe we can do it again. Take subjects from the general populations and do theta burst TMS to kind of create this virtual lesion, very loaded term. But really what we mean is we can use theta burst, which is a protocol of TMS, transcranial magnetic stimulation, essentially sending bursts of gamma, high frequency modulated by theta stimulation. And the advantage of that is it works a bit unlike the other, let's say 10 hertz stimulation or single pulse. It's not aiming to disrupt the ongoing task-related activity. It's not like when you're doing a task, I sat you right at this point. It rather works like a drug. So by sending you trains of theta bursts for, let's say, a minute, then essentially the excitability of the neurons in a region can be depressed for by up to an hour. So this is shown by John Rothwell, etc., in the motor cortex. So we apply this to the dorsolateral prefrontal cortex, the area that I highlighted earlier, and basically compare before and after, pre and post, in a visual task that is similar to what I described, discriminating a very simple shape in 2AFC, and then telling me whether you actually saw the thing or not. And surprisingly, Well, unsurprisingly first, it did not change the task performance at all. So my friends who think the prefrontal cortex, it does not matter for conscious perception, they're right in a sense. It doesn't change your ability to do the task. But if you follow the logic I've shown in the last slide, I wasn't expecting that anyway. I think the prefrontal cortex is important not for just performing the task. It probably involved, but it's not so critical. What is critical is for you to generate a subjective experience so that you can introspect and monitor what you're doing. And so here we developed a measure after a bilateral theta burst stimulation. So it's not concurrent because that would be quite dangerous in terms of seizure induction. But we stimulate one side and then wait a little bit and stimulate the other side. And then because the effect would last for an hour, so then in the next half hour or so, we know that both sides of your prefrontal cortex will be depressed. And then, so we created this measure that is a little bit of a mouthful to explain, but really conceptually, all it does is to assess the trial-by-trial correlation of your task accuracy and your subjective rating. That is, after a trial, I can see whether that trial you got the answer right, and I can also see whether you say that you saw the target. And usually, in a normal subject, these two things are correlated. When you say that you saw it, you're probably more likely to be correct. When you say that I didn't see it, you're less likely to be correct. So there should be a correlation. But this correlation, of course, is in some scale that is a little bit difficult to assess because there are many factors affecting it. So we apply signal detection theory to remove some of those bias terms, and then we put it in a scale that is easy to understand. So essentially, we're assessing how much information is available for this, what I call metacognitive monitoring, divided by how much information is available for you to do the task overall. So essentially, if you have like if you have 10 bits of information for doing the visual task, how much of that information is available for your metacognitive disposal of available for your introspection? So that means that this ratio, if it's 1, that means that you are basically theoretically ideal. All the information available for you to do the task will be available for you to monitor. So you see that these are really good subjects. They're kind of near optimal. But after you zap them for a minute and then you do the task, it drops by over 20%. And for those who do TMS, of course, a 20% effect size is not terribly small. So it's a decent effect. And it's not because people just got tired, because there's another sham control. So it's not just a session effect. Over time, people become less optimal. It's not the perfect control, but it's some control all the same. I didn't really believe the data, I would say, it would, because I mean, I believed it, but I didn't believe it very strongly. But over time, there was a number of good replications, some using very different methods, like muscle injection in animals, using very similar experiments. So I believe it's true. And I think I'm just going to mention one case is my good friend, Steve Fleming, also did a study that I think really add to some nuance. So he was actually interested in now that we have developed this method, is relatively sensitive, using psychophysics that would put people near threshold. He thought, well, okay, unilateral lesion patients may not complain that they become subjectively blind, but if you put them in this kind of similar kind of sensitive near threshold measurements, maybe you'll find something too. And lo and behold, he found an effect that is very similar to this on the order of 50%. So unilateral lesion prefrontal patients actually drop in this metacognitive measure by 50%. So they could do the task exactly just as well, but in terms of telling when they are likely to be correct or incorrect, they're all over the place. And that's amazing. 50% is a pretty big effect. And I think it's even more amazing is that he found that it's actually specific to perception, because you can do this task in a different domain. You can do this in memory. Ask people to do a 2AFC memory choice, old new. Actually, old new would not be 2AFC. I mean, old new or new old judgment. And then you can do the same subjective rating. Ask them, are you sure? And then you do the same measurement, and that measure did not change at all for the memory task. So that means that this prefrontal lesion effect was not specific, was not general about you just couldn't do this kind of complicated self-monitoring or introspection or do tasks or whatever. No, it was specific to perceptual metacognition, but not metacognition in general. All right, so this is actually quite interesting, I think. So this effect is linked to subjective experience, and it's not general about how you think about things in general. It actually is related specifically to your perceptual metacognition. I would argue it's actually probably because your subjective experience starts to be dissociated from how you can do tasks. So it's really quite specific to your consciousness. Now, why? Like you say, well, why the prefrontal cortex? What it's doing there, and how does it differ from metacognition? So for a while, I mean, I got a reputation. I would like to think, for the record, I never equated metacognition with consciousness, but many people thought I did. I was interested in metacognition for independent reasons, but I think now that it becoming clearer what's the relationship specifically between metacognition and subjective experience would be. So here's a kind of theory we've been working on. The idea is that, well, you might say, a lot of my opponents would say, well, when you consciously see something, let's say a duck, then you have duck neurons in your ventral temporal areas firing. And it might sound crazy, but actually sort of true. You do have neurons that are quite specific to animal categories. So you can have sort of duck neurons. They're not like grandmother cells. They don't fire exclusively to ducks, but they're selectively, preferentially interested in ducks. So let's say these duck neurons firing, then you would see a duck. Some of my my colleagues think so. But I think if you think more, I mean, it's more or less true, but it's not always true, is it? I mean, when you have these duck neurons firing, sometimes it's just because you are imagining a duck. You're holding the image of a duck in your working memory in order to match it to another duck or something, or to compare which duck looks more interesting or looks bigger. And when you do that, you also activate these very similar neuroactivities. They may not be entirely the same individual areas. I don't think so, but I think they're similar enough. Then the question is, then how do you distinguish them? And then, of course, sometimes you also have other situations when you have just spontaneous noise. So neurons are never completely silent unless the brain is dead or under very heavy anesthesia. So even just during resting state, your duck neurons are now kind of fluctuating every now and then and firing about threshold about mean now and then. But you don't hallucinate duck every few seconds, thankfully. So presumably somewhere in the prefrontal cortex or downstream would have to monitor this activity and decide what it represents. And I have to emphasize, this is not just a matter of whether it's bottom-up driven or top-down. Because think about when you're dreaming, it's also not bottom-up driven, it's top-down, but you see the thing is out there. And when you are imagining it, you don't confuse your imagination with external input unless you're in a psychotic state. So I think that's fair to be said. Like there's likely all these differences in schizophrenia and dreaming. The difference is actually probably even bigger in the prefrontal cortex than in the early sensory areas. I mean, there are differences in both, but I think the prefrontal cortex presumably play a role that is kind of like metacognition, right? This is basically monitoring a first order or sensory activity and trying to decide what it really represents. Is it representing the world right now, or is it just my own thing, or is it just noise? So this is a metacognitive engine. And there is a fair bit of empirical evidence that there is such an engine in the prefrontal cortex. And there's also a computational argument that you need something like this. Essentially, if you follow the deep learning literature, one thing that really blew up in the past decade or so is a method called generative adversarial network. So Sam Gershman at Harvard has written about this kind of length. Essentially, in order to have predictive coding, it will be advantageous for you to have this kind of engine to try to punish or compete with your generative network. So when you're doing this predictive coding, it would be good to have a kind of critic to say, well, your imagination is not as good, I got you, because your generation is so clearly different from the external input, so I'm going to punish you a little bit. So having this engine, which would serve as this kind of framework to stimulate the development of your predictive coding engine, and all of these are obviously very good things to have. So there's a developmental or computational argument why you want to have this, and there are also physiological evidence you have this. And in fact, there are single cell studies too, showing that you can distinguish between working memory content or perceptual content in the prefrontal cortex, single cell in monkeys. So I think it's pretty good. I mean, I'm pretty happy with the theory. I've written a preprint about it, and I also actually just finished a book over this thanks to the pandemic. The lockdown has done wonder to my writing regime because there's nothing else to do. So if you're interested, I'll be happy to share a draft of my book too. It should come out next year. But I'm not on a book tour. I'm actually want to talk about something quite different today because these debates, I think, have gotten quite frustrating in recent years. I think if you go to the empirical argument, I think we should have converged much much better already. But when it comes to theories, they're very loaded. And in this field, in consciousness, we are very invested in these ideas, sometimes maybe too much for our own good. And it becomes very sectarian, the debate sometimes becomes. So I got a little bit frustrated about that. And I feel empirically, I think we have the better argument, but sometimes it's not just a matter of sheer logic and experimentation. I think one way to think about this is to start to think about what applications we can develop and just go from there. And you can develop some applications, even my theory turned out to be wrong, then maybe that's good enough. And if you really think your theory is right, you should also try to generate some clinical applications. And if your theory is wrong, then presumably your applications will be no good. Then I think we will resolve the issue anyway. So think about just this very simple cartoon really model. We're doing some more computational modeling to make it more explicit, but just take this cartoon model, this idea that, okay, you have a duck representation that could be conscious or not conscious, depending entirely on a later downstream monitor. This already said something interesting, because this is quite different from another traditional view. I call it traditional because it may not be entirely dominant, but it's quite dominant view that your conscious and non-conscious sensory representations are in different parts of the brain. They're different channels. So at some point, stand there and call it a dual channel model, dual route model. And other people sometimes would think there's a dorsal stream is non-conscious and ventral stream is conscious. And other people might think cortical processing is conscious, subcortical is non-conscious. So if they're just different parallel channels, well, then it's quite different than here, because here I'm saying that your conscious and non-conscious representations are basically shared. They are shared all the way up till some very late stage monitor. And if that's true, that's interesting, because that means that any non-conscious manipulation of your sensory repertoire would actually also subsequently change your conscious experience to some extent. It's the same thing. So any kind of non-conscious weird clinical or interventions would ultimately change you. You don't have two separate one conscious and non-conscious process. In that case, you change this, you may not change this at all. They might interact, but they are not the same thing. Whereas if this is actually ultimately the same representations, by conditioning and rewiring your non-conscious representation, ultimately it will change you. So that is basically what I want to talk about today. Just one trick. I'm gonna focus on this, but also in the end highlight some possible extensions. So the idea is called non-conscious exposure therapy. So for those of you who are clinical psychologists, you might be aware that exposure therapy is kind of really one of our pride and joy, one of the things that really works. So the idea is based on decades of animal learning model, a lot of it done at Cambridge. So the idea is, let's say you're afraid of spider, then well, what you need to do is to gradually be exposed to spider, think about spider, imagine spider, look at spider from a distance, and eventually get used to the idea of having spider. It doesn't bite you really. So the idea is basically kind of like an extinction learning related inspired method. So your CS, you've somehow been conditioned to think the CS is bad for you. But if you just keep presenting the CS without any bad consequence over time, you would learn that it's not frightening. But of course, the problem is all of these are demonstrated in rodents and very powerful. But when it comes to human subjects, the difference is the human subjects do not enjoy it, and they have the volition to choose whether to continue the therapy. So if they do go through the therapy, I would say exposure therapy is fairly effective. Especially if you use some other tricks, I'll talk about later. But I think a major problem is people just often terminate the treatment if they seek it at all. Once they find out that I'm paying money to be told that, well, just hang out more spider, you'll get over it. They tend not to like the idea very much, and many of them actually walk out. In animal phobia, it may not be that big of a deal. In PTSD, it actually is a major reason. You tell people to try to relive your trauma, and you'll be fine. Many of them just walk out from the room and say, like, well, why don't you give me a pill instead? So there's an argument to be made. If we can make this whole thing non-conscious, so they're none the wiser, they don't even know what the purpose is, and they don't feel any subjective unpleasantness associated with the CS, that is the thing that they're afraid of, then it may work. It may work quite well. So that's how we started. So non-conscious, I think many colleagues here have different tricks, such as doing it while they're in sleep, while they're hypnotized, or you can use visual masking to just mask out the stimuli. I'm not so hot about masking. I used to be, because I think masking will give you a very small signal, essentially. Masking does not just abolish your subjective experience, it abolishes your entire perceptual signal, if not entirely close to big enough. So you don't have much signal to work with. I'm interested in the sleep work and hypnosis work, but I'm not an expert. But I end up doing something a bit funky because I went to a conference. In fact, it was one of those conferences Tris and I were both in. We were maybe not. I shouldn't say we. I was not in my top of form that morning, but a very senior Japanese scholar gave a talk, and I realized that it would be embarrassing if nobody showed up. So I, forced myself to show up in the morning talk, and then I learned something really amazing. So that was something that they did then in Japan. It's kind of crazy study. It was because over 10 years ago, it was really very funky. The idea, funky in a good way. What they did was they combined multivoxel pattern classification, which I'm sure many of you know. Basically, instead of just looking at the overall activity in a brain area, like those old school study I did, these days you can actually train a computer pattern recognition algorithm to recognize the fine-grained pattern of activity within a brain region. So any subtle change in the spatial pattern can reflect some content within the area. So let's say you take the visual cortex, which actually activates the red lines and green lines to a similar degree. If you actually train a pattern classifier, you can actually classify the two different. So if people are thinking about red lines or thinking about green lines, you can actually reliably decode which one is the case based on this fine-grained pattern. So what they did was to combine that with online, basically closed-loop neuroimaging. So what they did was to tell the subject, well, focus on your mind in some ways, and then now I decode your brain. And I decode some area, and then after about 12 seconds or so, I tell you, okay, well done. You did exactly what I wanted you to do. You got a big feedback, a big circle, and also that means that you got about like half a pound or something, a little bit of money. Probably not half a pound, not as generous, maybe a quarter of a pound or something. But so you got some money by based on this feedback, and over repetitions of a few hundred trials, that will be a few hours done over a few days. People actually learn to activate the pattern consistently. And then at that point, you might think, oh, they must have figured out the task. So they know that, okay, so the target, so what you're decoding me is actually trying to decode green patterns, so green lines. So every time I think about green lines, I get big feedback. You ask them whether that was the case. They actually said no. They actually said something kind of silly usually. They would say, well, either say, I don't have any strategy, I just relax. Some people would say, I have a strategy. Every time I think about Mozart music, then I would get this. I would humming Beethoven the whole time. Or if I was thinking about my girlfriend, hopefully your girlfriend does not look like some green lines. So clearly they are not, they have not figured it out. They're very far from having figured it out. In fact, if you ask them, in a forced choice, actually, okay, none of what you said was right. You are either in a group in which you activate green lines, you get big feedback, or you activate red lines, you get big feedback. Which group do you think you're in? And then they would, okay, they have to guess, and then they, as a group, there would be a chance. And that has been shown over 10 studies or so, that it's consistently a chance. And if you think about it, that makes sense, because here, you never told them what to do. So this is, in a way, not like neurofeedback. In neurofeedback, you tell people what to do, and you use the feedback signal to try to encourage them to do it right. This is really kind of like a passive neural reinforcement. You don't tell them what to do. You just wait for the spontaneous brain activity to resemble something. And if it does, then you just reinforce it. And as I told you earlier, your spontaneous brain activity is mostly non-conscious. So you're really reinforcing a non-conscious signal to make it happen more and paired with reward. So with that, basically, we have a kind of nice method to without complete unawareness, to induce specific brain representations in the brain in a pretty powerful way. So powerful that you can actually decode it well without chance using fMRI. So we did this crazy study. We presented these red and green lines to the subjects. So we learned these patterns, trained the decoder, build the decoder. So we now learn this pattern from each subject. And then we do this kind of very simple fear conditioning study or threat conditioning study, what we call these days. So let's say the red lines are called the CS plus and the green lines are called control CS plus. So both of these are CS pluses. Both of them have been paired with a mildly painful electric shock to the foot. So you present this pattern and you shock them, actually. So they become really, they get to really dislike and be afraid of this pattern. And then you also have a yellow pattern here, we call CS minus. It has never been paired with electric shock. So that's the baseline. So our plot next would be your skin conductance response to the target and to the control relative to the baseline. So unsurprisingly, of course, then both of these would be positive. So both of these bars are positive in the sense that your target CS plus, after conditioning for a dozen trials or so, your skin conductance would be higher for this and this than compared to the baseline yellow, which means that basically it worked. It replicated another thousands of studies. Basically, after peri-electric shock, when you just see the stimulus, you now show heightened skin conductance response because you are somewhat afraid of this stimuli. So we acquire the fear. And this is the central part that is interesting. We put them into the neurofeedback procedure I explained earlier. So you present these basically just gray lines, just a holder to tell them, focus on your mind. And every time you, if you do the right thing, I'll give you big feedback, but you don't tell them what to do. And after hundreds of trials, in fact, so actually the target, let's say the red pattern was chosen for this subject. Of course, it's counterbalance across subjects. But let's say for this subject, it's red lines. So every time red lines occurs in the brain, a representation of red lines occurs in the brain, not physically, then you get big feedback. So essentially, this becomes a kind of exposure or counter conditioning, because every time your brain non-consciously think of red line, not only that you don't get shocked, but now you actually get reward. So you kind of counter condition out of fear. The bad feelings of being shocked is now being compensated for by the stimulus representations being associated with a positive feeling of getting money. So lo and behold, in fact, it will reduce your skin conductance response after kind of a few hours of this training. Your skin conductance to the sorry about that. What's going on? Yeah, your skin conductance response to the target, but not the control, would specifically drop by about half after a few days. All right. So I saw some raised eyebrows, and this is time for me to move to the next screen and take. Do you have any questions right now? I realize I've been speaking rather fast. If it's so far okay, I mean, you guys are nice guys. I mean, when I give talks in some other departments, I've been like slaughtered right here. They'll say, this is too good to be true. It could not have worked. Clearly, it's a fluke, blah, blah, blah. Yes, the reviewers also said that. They grudgingly let us publish it because I think it was the first issue of Nature Human Behavior where they really just needed papers. So they kind of let us in easy eyes. They were like, well, okay, it worked. I mean, I know. It's maybe too good to be true. We will try to replicate it later. And then we succeeded in convincing them. But we did replicate it later. That's a good story. So Vincent, another postdoc, so this last study was done by Aiko Izumi, and Vincent Testro Dumochel came to my lab, and I thought, well, we'll replicate it. But Vincent said, well, that would be boring to just replicate it. Why don't we take it a notch further and do something more exciting and actually try doing it with real life stimuli? And of course you can do that because decoding real life stimuli is not difficult these days and with today's fMRI technology. So we did a task that is pretty simple. We present images of different things like snakes, spiders, something scary, and something non-scary like rabbits, etc. So the task was they see a picture every second or so, and then once they see a category change, they have to press a button. So they see snake, 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 and then you change the rabbit, and they press a button, and they see rabbit, rabbit, and then you change the dog, they have to press a button. So whenever the animal or object category change, they press the button. So it's basically just to keep them attending. And we show a lot of images from many categories of things. So then we learn to decode the brain. Now the problem came back. We thought, okay, then we do this and we replicate the last study, then we can actually counter condition out fear for spiders or snakes that people are afraid of. That would be nice. But then we realized that we got into a kind of logical problem, because why do we do it for real life stimuli? Just, I mean, one idea just to be cool, but ultimately Vincent has a clinical background. He said, I actually want to take it to clinic to actually help people. But if you actually want to do that, then recall that the logic has been, well, the advantage of this, having spent thousands of pounds on each subject to treat this trivial problem of phobia, maybe not so trivial if you're really severely phobic, but the promise was then I don't have to consciously see these things, right? It would just be non-consciously happening in my brain. But in order to decode, to build a decoder for spiders and snakes and all this stuff, you're showing me these pictures. Then, of course, the patients who would drop out from these studies would drop out at this stage. So it's actually not going to work. So it kind of came back to square one. But Vincent came up with this really, I think, clever solution. I mean, I would like to say that we invented the method. We didn't. We stole it from Jim Haxby's lab. He already presented the solution before we had the question. But I think we're the first one to came up with an actual clinical application for the method. The idea is called, the method is called hyperalignment. So the idea is, if you, let's say you're afraid of snake, but you don't want to see snake in your treatment study, then, well, you don't have to see snake. You can just see apples, orange, dogs and cats and rabbits and whatnot. And I would see also apples and oranges and dogs and cats and want to see the same thing. So after you and I saw the same thing that doesn't include snakes, we can try to rearrange spatially our voxels so that we will be basically in the same space. So that my dogs and cats and whatnot would be same as your dogs and cats and whatnot patterns. So our patterns are not supposed to be by nature the same, because it depends on our fine-grained vascular structure and organization. But once we have these other stimuli that we've both seen, we can use it as a basis for calibration. We move our voxels so that spatially they're now in the same space. So now that I know that my brain in that analysis will be in the same space as yours, then now I can see snake, because I'm Cantonese, I'm from Hong Kong. We eat snakes, so I'm not afraid of snakes. I can just, watch a lot of snake images all day, and then I would get what my snake pattern would be, and then I would become your surrogate. So my snake pattern can now be used to infer what your snake pattern would be, because now our brains are calibrated in the same space in the analysis. And now when I first pitched it to my clinical colleagues, they raised eyebrows and said, yeah, it sounds cool and a bit sci-fi, but it must be very, very noisy, right? I mean, you can even do it in principle statistically about chance. We're talking about clinic. We have to get it to work well, not just working like a statistical effect. Then we realized one thing, because once you can have one surrogate, you can have 100, because I can see all these different things. You see the stimulus once, and I see all the other stimuli, and I can be a surrogate. I see snake for you, and I use my pattern to infer yours. Once you do that, you can repeat this procedure with another 100 other surrogates. In fact, we are now hitting 100. And so we have a database of a lot of snake patterns, and then we can average them, leveraging the power of sheer volume of data. In fact, now we can infer your snake pattern without you ever seeing snake very well. Actually, better than if you see the snake images yourself. Okay, so that was Vincent's study. And so lo and behold, this is the logic. We are mostly focusing on ventral temporal areas. We decoded those stuff from these, build those decoders using this hyperalignment procedure. And the logic is, for each subject, we pick two things that you're afraid of. So let's say for an average person, quite often will be snakes and spiders, but we actually tailor it to each person. Some people are afraid of caterpillars. Some people are afraid of cockroaches. We just pick two things that you're maximally afraid of, and we pick them, and then we randomize them in the computer and choose one as the target, the other as control. And this time it's fully double-blinded. We don't know which was which. I'll come back to this point later. It's quite interesting that it's double-blinded, in fact. And then one of which become the target. And then so during the neurofeedback procedure, that pattern for one of these things, we don't know which was which, get rewarded with money. And again, it's the same procedure, a few hundred trials over a few hours, spread over a few days. And now the question is, what would happen? So lo and behold, it worked beautifully. I took my breath away a little bit. I really didn't, I was hoping maybe we'll be lucky to replicate, but actually replicated, the effect was stronger still. So this is skin conductance. Very similar to what I assume is shown in her study. So this is actually before and after, and this is for the target. You see that for the target, it completely basically went flat instead of reducing by half. And then for the control, it didn't change at all. So it's really specific to the target. It's not just like generally people get, your skin conductance become weaker signal. And also we're showing here is your amygdala bold response, which can also be taken. Some people take it as a reaction, kind of your excessive fear response in the brain. And again, you can see that basically flattened, if anything, almost went in the opposite direction. I think it's not very significant, but the difference is definitely there. So basically flattened all your physiological responses. I thought this sounds great. We were so excited, and we published the paper. But again, the reason I give these talks is I kind of come to confess that we cheat a little bit. You don't see these in papers, because in papers you need to be perfect, but in talks I can be more honest with you. There was one disappointment that I didn't put in the paper. We debated about it. Should we put in the paper? Yeah, well, maybe we can. We subsequently did publish it in a second prepaint, but in the first paper we didn't mention it. Maybe that was a little naughty, but that was the disappointment. And it's quite interesting. Is that, well, when you ask people before and after the procedure to actually look at those images, because these are not clinically diagnosed patients, they just have a normal range of fear. Like, I'm very much afraid of cockroaches, but I wouldn't call myself a patient for that reason. So these people could actually look at the image. They can tolerate that. And you ask them how much afraid you are of the target and the control. You can see there's not 6. 6 would be, I think 6 is defined as the level at which if you see this image, you might have to terminate the experiment. So it's not that bad. 0 would be nothing. So they're quite afraid of these things. That's why they're selected as target and control. But you can see before and after the procedure, the ratings did not change at all. So a change of physiology, flatten your physiology entirely, but subjectively, if you ask them whether you're afraid, they said it's the same thing. I'm just as afraid. So it's kind of like a downer, but it made my colleague, Joe Ledoux, my friend and colleague, very happy, because he actually wrote books which actually predicted this before we did the study. He actually has been claiming then that the amygdala is actually not a fear center. I mean, he's often been ascribed with that idea. And he said, well, no, I never really meant that. What I meant was amygdala was important for driving your physiological arousal, or like what Dominant Bat would say, maybe the prediction error related to threat-related processing, but not the subjective experience itself. The subjective experience itself can be completely dissociated from this physiology. So that's a little bit of a downer, but in a way, it's a good result, because think about this. So I earlier talked about how this late-stage monitoring idea may apply to vision, and I think that maybe not a done deal, not a proven theory, but I think it's fairly likely. I think it's true. And the same might apply to fear. So in fear, you have an amygdala and maybe your insular signal that basically reflects your physiological arousal, the excessive arousal related to threat. But it's not fear per se, because for you to experience fear, you really need the prefrontal cortex. Again, your prefrontal cortex needs to recognize that this excessive physiological arousal signal reflects something that is happening to you right now. It's not your imagination, it's not your empathy when you think of other people's fear. Is something that's happening to you right now, and that's where you have this subjective experience of fear. And Joe Ledoux and I have been since then writing some papers together to develop this further, and so did Richard Brown. Now then, this is actually a controversial idea within the fear or threat field, anxiety affective studies. Many people do not agree with us. I think part of it is because many of those people work with rodents, and for many years they've been arguing that studying the amygdala is the holy grail. We can via that treat all the anxiety disorders. And Joe was part of that, and Joe came out and said, "Well, I don't think it actually worked. I mean, we spend a lot of time and money investing into this, and people are actually not completely cured." And so it's because of controversy, it would be good to do a study to further test the idea. And this will be my last study I'll show, so don't worry about my overrun too much. So here, Vincent actually didn't do a new study, but analyzed the same data. So this is from the same data, and you can actually look at both the SCR, because we recorded skin conductance, we also have subjective ratings. You can actually plot the SCR against the subjective rating for these categories. You can see that there's a positive correlation, meaning that the people who, the categories that show higher, that induce higher skin conductance are also the thing that people are afraid. So the two are correlated for sure. That's why people use SCR to study fear. They're not being wrong about that. But you can also see that the correlation is not completely perfect. I mean, some things are more like this spider here that drives more skin conductance than subjective fear, and some things are subjectively more fearful, but that didn't drive you nearly as much as skin conductance. So there's a correlation, but not a perfect correlation. So capitalizing on that, that means that we can try to build brain decoders that might selectively predict one but not the other. That is to find maybe there's some regions that you can build decoders based on activity in those regions that would predict better skin conductance rather than subjective fear. And you might find other areas that might predict better subjective fear rather than skin conductance. And lo and behold, it in fact confirms what we suspected. All these areas in amygdala, insula, ventromedial PFC, this traditionally considered kind of like a fear network. In fact, those areas really predict skin conductance really well, but it doesn't really predict your subjective ratings of fear nearly as well. Whereas if you really want to predict the subjective ratings of fear, in fact, they're all in the lateral and anterior prefrontal areas. Those are what really predicted your subjective ratings of fear. And so this confirms kind of the idea that Richard Brown and Joe Ledoux has been writing about. And it also addressed one point in consciousness research that we care about. Because if you think about it, this is actually a kind of a no-report study. In fact, it's better than a no-report study, I would say. Because here, remember that during the task, people were not rating fear. People were just pressing a button when they saw the category change. The fear rating was done outside the scanner later. So we're decoding the fear response, presumably the fear response, associated with images that are subsequently going to be rated as fearful. But they're not rating the fear during the scan. So what I decoded could not be the act of reporting or rating. It's decoding the experience that we infer based on subsequent reports. So there's been some claim that from the prefrontal cortex, all you decode was just the report or just the ratings. I'm sure that is not true. In fact, if you're interested in this, you see this review. There's been a lot of studies that have already taken care of these no report confounds, well before the buzz phrase no report becomes nowadays so popular. So it's okay to control. I mean, more control is better, but I think it will be kind of misguided to think that this will be a new kind of control paradigm that will lead to new results. We're taking care of no report for over a decade, actually for decades. So it's not a new thing. It's good to do clean experiments, but if you think they would lead you to some new insight, you might want to think more and read more of these kind of papers that already taken care of the control. So now you may think, the good thing now we have targeted PFC patterns, so we can then try to see these reflect subjective experience. So we might be able to do this study to change your subjective experience. And then by doing this neurofeedback, so we can now not just flatten your physiological response, but actually change your subjective fear rating. I'm going to give you a downer reply that actually we're not quite there yet, because we have to distinguish two things. What I showed you is your prefrontal cortex can be used to decode your subjective ratings of fear, but we couldn't decode quite well your animal identities actually in prefrontal cortex. That is, you can decode your level of fear, but for the study to work, you need to find prefrontal representations of specific animal categories. At some point, we thought we kind of got it, but actually there are some overfitting issues. So actually, it's not quite easy to decode content from prefrontal cortex yet. You can decode some, slightly above chance, but it's not really good enough to be worth putting in the clinic. But we are putting in work in progress because we have a lot of data on this. So with the method changing, I do think that eventually we might get there. But regardless, somehow we have managed to convince NIMH to fund this for a clinical trial. So we actually, as we speak, had it not been for the pandemic, we actually already tested a few patients, but had not been for the pandemic, we might have been close to completion. We are actually doing this, focusing on ventral temporal areas. That is the what we did in Vincent's study, not the prefrontal cortex. And we are actually doing this study to try to see whether we can help patients. Now you might think, why would it help? You already shown that it only flattened the physiology, but not the subjective experience. So one argument is, well, maybe with time, the subjective experience also would change. Or maybe it's just a dosage effect. But I think even if we really don't change the subjective experience, but just the physiology, I think it already helped a lot with the attrition problem. Because if you're physiologically, even if you're afraid, you're physiologically not so over aroused when you just think about or encounter these things, it might means that it's much easier for you to go through your psychotherapy, because people don't like the idea of going through it, true, but also some of them just faint or get really physically sick doing that. So like dampening the physiological excessive arousal might help. And the other issue is relapse. We know that in exposure therapy, one problem is they learn that, okay, this is safe to hang out with the spider in this room in front of this experimenter. But as soon as I go back to a different context, there is contextual renewal and all that. But here, I think the advantage is we might not have this problem because we are directly treating the brain representation in the brain. And that should be not context specific because it's the same voxels anyway, regardless of what context. So it might generalize better is a hypothesis we're testing now. And finally, I think this part is important for nerds like myself, probably not for the practically minded, but I always feel that clinical psychologists, we train a lot of them in my department. And why are they not considered like real doctors, right? They're sometimes not considered like mainstream medicine. And we know the insurance policies are different. And sometimes they're called evidence-based medicine, but not like real mainstream medicine. Why? I think one of the reasons is just because of this double-blind issue, right? So since the 1970s, all of internal medicine, now the vaccines and all that, it all has to be tested for so long because they have to be double-blind, placebo-controlled. And when you think about clinical psychology, that is just impossible. There is no placebo treatment. You can't do talk therapy in a placebo way. That would be kind of weird. And even if you do, you can't fool the experimenter. You can't fool the therapist. The therapist giving a sham talk therapy presumably would know that it's sham. So and the level of enthusiasm would be different. So you can't really do double-blinded stuff in clinical psychology. If you know of a counterexample, let me know. I would be curious. But I almost feel that in principle, you can't do it. But here, you're taking the rationale of clinical psychology and testing it directly in the brain in a completely automatic, non-conscious manner. So it's actually double-blinded. If you actually got this, I'm going to take this to the doctors and say, well, this is actually now medicine. So this is not clinical psychology. You have to include this in the same status, and we'll see how it goes. So I'm about to wrap up. We're still doing more studies, and this is actually so far kind of interesting, but we don't know how it generalized. I hope that it generalized to other things. So we've been trying to use more realistic animation and decode more than just animals. We can decode car crashes, being pointed at, having a gun pointed at you by a policeman is quite traumatizing. Or some people might just have social anxiety. So all these like different things you can decode in the brain. And we're also doing a study now on pain, on chronic pain. So pain is very easy to decode in the brain. If you let me just shock you, your whole brain kind of lights up, and I can find these rather widespread pattern distinguishing between pain versus no pain. So here the idea is not to just down-regulate this pain activity, but rather we got your pain pattern and then we reverse it, and then got this no pain or anti-pain pattern. And then what we do is in the scanner, we try to pair it with an arbitrary visual stimuli. And because so far we've been doing this associative learning, it seems to be quite powerful. And imagine if you now eventually pair this image with the anti-pain brain pattern, then it's actually quite good because you don't have to be in the scanner to feel no pain. If you just happen to have a really bad pain, then you can just take out this postcard and look at it or imagine it, and that might actually activate the anti-pain pattern. So that could actually have a better clinical potential. And further, because the pain pattern is not so fine-grained, you could possibly also do an EEG, and we haven't planned doing it yet. If you want to steal the idea, feel free. I'm very keen to have more people doing this kind of work. And this is just finally a take-home kind of point, almost like, so with Jack Gallant's work, I think he did some of the best decoding work. And this one kind of took my breath away. He actually just read stories to people in the scanner. And with that, he created this image of what's called a brain dictionary. So there are different words, different concepts in the brain that you can actually decode. So your concept of power, parenthood, lust, money, fame, all of these kind of, very, very touchy-feely concepts can all be decoded in the brain. So imagine you could actually associate these with different things non-consciously. Essentially, all these like Freudian, psychoanalytic, lofty goals may not become so crazy after all. Maybe you can actually manipulate all of this. So let me wrap up. I'm overrunning a little. Let me end with this quote. This is from B.F. Skinner. Of course, we psychologists have a very, very complicated relationship with this guy. Towards the end of his life, he actually wrote about consciousness, and he turned out to be a kind of closet Freudian. But this is catching him in a more reasonable moment. He was defending behaviorists. I think this behaviorism, I think it's the best defense he has given. He was saying that basically no major behaviorist has ever argued that science must limit itself to public events. That was a bit of a shock because I thought that was the whole point of behaviorism. But he went on to clarify and said, well, as a behaviorist, however, I question the nature of these private events and their role in the prediction and control of behavior. So you say that, well, of course you can study private stuff and all you want, but in Pavlov, we have this beautiful method through associative learning, through all these Dickinson models, and we know how to, basically the models of associative learning is so powerful that with that, we know how to predict and control behavior, all these applied behavioral analytics and all that. And if you want to talk about consciousness, well, good for you, but probably your models would not predict much, and you would not lead to very well controlled interventions. And I hope I want to show you today is, well, you can have the cake and eat it. I use the Pavlovian methods like you did, but I also talk about consciousness. And if you combine the two, maybe you can even do better still. And I highly recommend that. So thank you. Thank you very much.
Thank you very much. You can clap however you like in the system. I prefer to clap, physically. I open the virtual floor for question, comments, criticisms, dances, whoever wants. What's going on? You can also write in the chat if you're initially shy. And I can ask a question.
Raise the hand or something. What's the system?
Whatever it wants, I'll manage it. If someone wants to talk, go ahead. Open the mic and chat. I'll manage.
You're gonna stop sharing? Yeah, you can stop sharing. I see some question in the chat.
Yeah.
Oh no, they're not.
No. I'll tell you if there are questions in the chat. But
Rick Hansen. Hi Rick, long time no see. You have a question.
Hi, thanks for a great talk. This may be a stupid question because I don't know a lot about behavioral therapy, but so you're using this neural reinforcement to associate the CS plus target with a positive outcome, i.e. money. Does that kind of new association transfer to reduced physiological response to the CS plus? So you showed that it reduces galvanic skin response, but would that be expected? I mean, these seem to be two quite different things. I could associate a spider with money, but still be very fearful of it. And so in other paradigms, and in behavioral therapy, I don't think you show people, expose them slightly to a spider and give them money or give them some reward. You just show that that doesn't result in any negative outcome. So I guess what I'm asking is, I don't know enough about this field, but is associating it with a positive outcome the same as actually reducing the negative outcome, the negative autonomic outcome of galvanic skin response?
Yeah, right on. They're actually not exactly the same. So I kind of gloss over a little bit. When I started out, I was inspired by exposure therapy, in which you just expose, right? You just do extinction learning. So in the animal learning literature, there's I think Bhutan, is a person who have done systematic studies and showed that counter conditioning and extinction are different. They're different mechanisms actually. So we're actually tapping on a slightly different mechanism here, but there are two things. One is it might be good to compare systematically in the clinic whether exposure and counter conditioning, how it would work differently. But I would think normally in the clinic, even though the exposure therapy is sort of inspired by extinction, that is just presenting the CS plus without a negative outcome, I think in the clinic actually mostly work more or less like a counter conditioning because of that pat on the shoulder effect. Because your therapist, when you see, you can now see the spider without you've been doing so well. So the social signal actually might work out about the same.
Yeah, that sounds plausible. Yeah.
But certainly we should do more studies and figure it out.
Thanks.
Any more question, comments? I see people are coming in. Andrea.
Hi, thank you for this super fascinating talk, Hakwan. It just, just one of the questions that popped into my head early on was when you were talking about the hyperalignment, and you have these patterns that are individual, but at the same time can be predicted from these other people, so that you don't need to show the snake to your patient, but you can just show the snake to someone else and infer the patient's snake pattern, if I understand correctly.
Yeah, that's right.
I was wondering if the effect that you are observing in your studies could be predicted by the similarity of the patient's particular snake pattern to some other kind of patterns that are perhaps associated in this third way with, in a non-immediate way, with fear or other kinds of stimuli. This was prompted by the Gallant lab picture that you showed earlier.
I would think so. Yes, I would think so. So in fact, that if you really dig deeper, we've been thinking about this to make this really practical. In fact, there could be a way that we ultimately don't need the scanner in a sense. If I can just figure out your snake patterns are very similar to these other 5 animals, or the combination of these 5 things, of which you are not afraid, then maybe I can just expose those 5 things to you, right? Because it comes down to the same brain pattern. So that's an interesting thing to think about. We've been thinking about it, but the short answer is I think you're right. But maybe you're asking a question. Probably you mean how to exploit that, and we have to think more. Yeah.
Thank you.
Any more questions, comments? I have like 5 things in my list, but I don't want to do the host questions.
I'm very glad that Tris, you set up the hostile environment and still no one comes to challenge me. So I hope that I probably silence my critics today.
I'm sure that I will get emails about it. You know.
Okay.
I had a follow-up to my question, if that's allowed.
Yes, please.
Yeah.
This is probably stupid, but
No.
why? So this issue about whether counter conditioning works the same as extinction. Is there any reason why you couldn't run the neural reinforcement with a negative feedback, a shock, rather than positive outcome like money? So you basically omit the shock if the neural pattern is resembled CS plus by chance.
Yes, I think I get it. No, it's not super at all. It's actually a very cool idea. We didn't quite do that, partly because I think we could have done it, but the reason we didn't choose to do it, so because when you punish the pattern, then if it was so effective, then the spontaneous occurrence of that pattern the chance would diminish. So you would reduce the chance of being able to further punish it because it kind of basically shies away from it. And so we kind of chose the more positive way. But the fact of the matter is, we could still try it in the future because the punishment is not so effective that once you show this pattern, you get a pair shot, it never shows up again. I wish it was so effective, but it wasn't. So in fact, it's worth a try. We might do so in the future. I think people react to, because of the money we give them is not £1 million. It's just a little bit of money. A shot might be actually more viscerally important to them.
Thanks. Go ahead, Tristan.
I think Deborah was. She went back into mute.
I think Alessandra has a question.
Oh yeah, Alessandra, if you were, maybe not.
Okay, I don't have exactly. It's not exactly a question. It's just maybe a supportive comment, if I may. So I think you're right to say that it's highly likely that patients' subjective response to the fearful stimulus might change with time, because their understanding of people's understandings of these semantic categories is very strongly established. And it seems very likely that they use their explicit knowledge of semantic categories to tell you, yes, I am the person who's afraid of spiders. That's not going to change without more experience. And it reminds me of a finding that we're writing up at the moment, where we're asking people to rate the similarity between emotional categories that are not similar to each other semantically. So say begging on the one hand and car accidents on the other. So there's not a strong relationship. People say that they're not related to each other particularly, but in terms of neural similarity, there's really strong similarity between these categories. So we see a dissociation, and Joey Dunsmore finds the same thing, that the similarity between CSs is greater in the brain than the similarity than the subjectively reported similarity. So I think we're seeing something about the resistance of semantic categories to very quick changes.
Yeah, I think totally agree. I think that's right. And we are now also doing this follow-up in patients. So we would instead of just subjective rating, we also do behavioral approach test. So we will see whether they actually are more willing to approach the object in virtual reality, for instance. So maybe I think you're right. It's just that people's mindset is already fixed. If you ask me whether I'm afraid of a cockroach, I would say yes, even though I'm not going to imagine it or anything. But if I go to see a lot of cockroaches, hopefully never, then I would probably find out, I'm not jumping anymore. And then I may over time learn that I'm not afraid of something. So it might take a bit of restructuring of the semantic process. I think we agree.
I have a comment for you to maybe sort of comment on top, which is I was thinking about the classic textbook thing of HM, where HM came and they shocked him with a by giving the hand, and then the following day, etc., etc. Unconscious knowledge of of something negative, etc., etc. I'm not sure if there was any post discussion with him after other days, whether he actually became aware later and made the connection. He definitely had the unconscious learning part on a one trial association. How is this classic aspect related to to what you're saying in patients? And because my feeling is, again, I agree with you. I don't care that much that a person has the same level of subjective rating initially. But what do? You do like the therapist. You convince the participant you're not afraid anymore. And then they believe themselves. I can see objectively you are not afraid anymore. Or you said you don't have the response that I expect from someone who's afraid, but you're still saying you're afraid. Convince yourself. So where do we go from here? Almost clinically.
Yeah. I think that's absolutely right. So in the case of HM, I think is trickier because he's the hippocampal damage might mean that he couldn't really relearn. So even if you realize that, oh, now I'm very afraid of this person, but he may not come to restructure his thoughts enough because of the pathway being somewhat damaged. But I think for the normal subject, I think that's right. I think over time with either a good prefrontal cortex or a prefrontal cortex which we can mess with, I think they would ultimately come to learn to convince themselves, I'm actually not afraid of spiders anymore, because and then with time maybe even the subjective ratings or subjective experience would change. Does it make any sense? So having flattened the physiology, and in the old literature, something they call it the synchrony between the three measures of fear related stuff: the avoidance behavior, the self-report, and the physiology. And in the old literature, in fact, there is some discussion of that synchrony too, but it never properly. I shouldn't say not properly, but not having been heavily investigated. Somehow, kind of people just went with the physiology and become really fixated with it. But I think there's an opportunity for us to now see that this disassociation, what can allow the two measures to be more synchronized. So perhaps, like for instance, doing a TMS to the prefrontal cortex might allow them to recalibrate in a sense. So the view I take is the prefrontal cortex is monitoring these physiological signal and thereby deciding or generating a subjective experience. And one way is for the prefrontal cortex to be really clever and just learn, okay, I'm not afraid anymore. The other way, maybe you can kickstart the process by basically sending TMS pulse to prefrontal cortex and breaking that link, so to force it to remonitor, rebuild the monitoring relationship. So we've been thinking a lot about it, but I don't have anything more concrete to say. It's a great question, challenging.
There's still 40-something people around. Who wants to ask a question? Don't be afraid of Hakwan. He has proven to be nice so far.
I tried to be nice today. I kind of slipped a little bit in the middle, but I reminded myself I should stop.
What is this? What is the relation between these ideas and the classic hypnosis therapy for fear, where the person is not supposed to be aware when they are when they enter in hypnosis? You can convince them, if they are allegedly well hypnotizable, that whenever they encounter this, they won't feel fear, or they won't have the response of fear. So this is where the tricky dissociation could happen. Have you thought about this thing?
Yeah. I think I'm not an expert on this, but I know some of this work from people like Sultan Dianis and colleagues around in the UK, and also Talia Whitney has done some work on this. I think my take is when hypnosis really works, is wonderful. but it doesn't always work for each subject, right? So the suggestibility differs. So we can think of this as just a complementary or similar way of doing similar stuff, but it works for most people. So most people have spontaneous brain activity and have these non-conscious processes. So in a sense, it is logically similar, but this method might be less, maybe more generally more applicable. Although Sultan also has a finding that if there's a way to increase people's suggestibility for hypnosis, which is to give them beer. So it turns out people after a pint will become more likely to be hypnotized, but I still think some people just are not hypnotizable, which is a lot of them.
Can I just jump in? I couldn't find a way to raise my hand. Hi, Hakwan, it's Anil.
Right.
That was terrific. Yeah, I was thinking along similar lines of Tristan actually, because I'm beginning to work more with Zoltan and Pete Warwick on what they now call phenomenological control rather than hypnosis, because of the baggage that has. And I think you're absolutely right that there is this strong but quite stable individual trait difference in suggestibility and ability. And it really, I couldn't quite formulate the question, but there does seem to be a strong, not just they're complementary, but perhaps some strong overlap here where those individuals that are high in phenomenological control or high in trait suggestibility are those individuals able to generate sort of subjective experiences through suggestion. And importantly, the suggestion doesn't have to be like a conscious instruction to experience X. I mean, it can be an implicit response to demand characteristics, which is all the kerfuffle that we've been having over the rubber hand illusion and how to interpret that. And it's not that just the context of the experiment can lead people to have a rubber hand experience more so than other people, even when it's just implicit truth. So I'm just wondering whether, very basic level, your methods might be predicted to work better for those people who were highly suggestible. If it's a sort of, especially if it's the DECNAF thing, the kind of intensive imaging.
Yeah, I think there's a solid hypothesis there that possibly, so after we flatten the physiological excessive arousal, some folks would probably, I mean, it looks like it basically didn't change the subjective experience at all, but I think Debbie also pointed out, this is kind of what you expect, but if you give them more time and make them actually interact and reflect and we didn't tell them the purpose of the study. So we already miss out on that placebo component, which is a huge part in any intervention. So if we actually go to the clinic, we'll tell them, actually, I tell you what, I just flattened your physiology. Now think about it. And then tell me whether you're afraid. Maybe that already would change a lot. But then I think, yes, just the degree to which their subjective experience would catch up with the physiology probably would be modulated by those things you just said about. You just talked about like this hypnotizability, phenomenological control, individual differences, etc.
Yeah, no, that's interesting because, of course, on the other hand, when you're presenting these things completely under the surface of awareness, then and they don't know what the context is, that could even be a counterexample about, okay, these things are going on independently because there's not even any implicit reason for them to expect certain things to happen, whether they're physiological or subjective.
No, I was saying that if you unmask the intention,
yes.
If you now tell them, then I think the how fast they would catch up to the subjective experience would catch on with the physiology would probably be modulated by those things you said.
Yeah, cool. Okay, thanks.
A small subject sample size, so right now we're not really quite prepared to assess individual differences yet, because these studies are quite involved for each subject. But eventually, hopefully, we'll get.
On the other hand, you could, I mean, if for the goal of sort of clinical application, I mean, what you could do is sort of do it the other way around and pre-select your sample. So you could take samples of highs, which you can assess online, and it's pretty reliable, the stuff that Pete and Brian and Zoltan did. Then you that would be a nice sample to think about clinical efficacy.
Definitely. Yeah, all this personalized medicine is all the rage, so we are definitely on the same bandwagon too. In my grant proposals, I always say that, but we haven't got to do that yet, but we will hopefully. Yeah.
Okay. I have an anonymous question that I just received. Anonymous. Yeah, I'll read it to you.
Okay.
Anonymous, yeah. Is by someone who doesn't want to be named.
Okay.
Thank you for your interesting talk. I have a couple of questions. One, how far along is the VR study? Two, in light of recent social developments in America, particularly the increased saliency of police brutality, both in the media and in real life experiences, how might that have factored or not in the decision to use that particular scenario, the one with police? And do you foresee any potential findings that might be derived from with this particular study, which will add to the existing understanding of the interface of identity, individual differences, and social cognition?
That's a great question, actually. Yeah, I hope. Yeah.
Both. So how far they VR, and then
yeah, not very far at all. We started piloting. We created the stimuli. In fact, some of these stimuli are just from the reason we chose some of them. They are from a game that some of you might have been familiar with. This game, Grand Theft Auto. So it's just a pretty popular and somewhat violent video game for adults only. It's pretty. So it has a lot of these violent scenes, and we just took it from them and I think that's right. We created a stimuli, we scanned a couple of subjects and try to decode these things. It looks like they are fairly decodable, but I don't have anything ready to show yet. And about the social unrest, yes, I think that example might, in hindsight, maybe a bit sensational. So I think the issue is really, it's quite an expensive treatment, even when it works. And in the pain case, we made a case maybe you can do it in EEG, so it could be cheaper, but it's still quite involved. And so every time we think about the issue, and likewise, early on we've been colleagues have suggested collaborations like why can't we do racial bias training this way, so we can decode the face of like as an Asian like myself, and if you happen to be racist against me, then I would just reward my own face with a lot of money, then you'll come to like me and that sort of stuff. Yeah, it might be good to show and would be exciting in some way, but in some sense, those are not serious clinical problems. They are more societal problems. Maybe there are ways to treat it more effectively via public discourse. This is quite an involved procedure that might be more suited for really otherwise untreatable clinical problems. As to whether racism is one, I should leave it for further discussion. I feel racism is a huge problem, but maybe not the same nature of this kind of clinical problem.
Good. Any more questions, comments? We're a couple of meters away. Oh, Peter is coming.
Hey. Sorry, I must confess that I got so excited at certain points in the relationship with my research that I actually lost some pieces of your talk. However, yes, due to low working memory and high excitability. I just wanted to generally challenge the notion that if you are not conscious of a specific stimuli that you are somehow associated or experiencing, that you will not have a negative subjective experience. Does that make sense?
So you mean the stimulus has been paired with something negative non-consciously, and then when you see the stimulus consciously, you may not think negatively of the stimulus. Yeah.
Well, say the stimulus that you're afraid of is subliminal. Mm-hmm. You don't know that you've seen it. Still, you might have a negative subjective feeling, and therefore the drop out.
Got it.
And like even yeah, exactly. And it's possible that that subjective feeling is non-conscious as well, but will lead to certain behaviors.
Yeah.
And hence, and also another notion is that the pattern, say you're looking at this, rewarding a specific pattern of the negative stimuli in a specific region. Potentially that same stimuli might be re-represented through other pathways throughout in the brain. And hence rewarding a specific cortical pattern might not do the trick.
Yeah, I think you're right. So two things. So first about the bad vibe issue. So in the beginning, we thought about it too. So when you look at during the neurofeedback or neuroreinforcement procedure, when let's say the spider pattern occurs, turns out those are not accompanied with spin conductance response, as far as we can observe, at least. So the bed might be there, but it's small, seems to be, because maybe these fluctuations are also small. So the other question is about the specificity of where do we target. Yeah, I think, maybe it's fortunate and unfortunate. The fact of the matter is we actually took the entire ventral temporal stream that has these representations. It's quite a large swath of area. So we cover a lot of voxels, but in the end, the selection of voxel is actually more driven by practicality rather than our model of how it works. Because in the prefrontal cortex, it's just very hard to decode any of these stuff. So you're right, maybe those areas will have some other functions, and maybe V1 also will have some other functions. But really, we just pick the voxels that work and take the whole lot of them. Doesn't mean that they don't exist elsewhere, but those are not so easy to decode. So we just chose the lazy approach, and let's not worry about them for now anyway.
Actually, I was thinking also of subcortical pathways, that we know that subliminal stimuli might have subcortical effects, etc.
Certainly they will have effects, but whether we can decode the identity of the animals from there is a trickier issue. And the cortex is very nice. It's like a piano. It lays out everything spatially. It's very nice to decode. Once you get to the prefrontal or subcortical areas, things are a little mushier.
Yeah. Thank you.
Hi, I have a question as well. Thank you so much for the talk.
Thank you. Nice to meet you.
My name is Amber. I'm just wondering the phobia possible treatment that you've just talked about, because I think that the experiments use the images. Do you think people will actually be less afraid when they see the real thing, which actually moves?
Yeah, that's a great question, and the hand wavy answer is it's an empirical question. We have to find out, and that's true. But I think I have a good hypothesis that it would work. The reason is actually in some sense, we capitalize on the bluntness of voxel response, because yes, we are reinforcing voxels, the decoding of which we learn from those images. But at the voxel level, I think it forced a generalization. That is, when you see a real cat, or you see the image of a cat, it's of course a very different experience. But in terms of the brain response at the fMRI level, at this resolution, they're probably quite similar. So in that sense, the lack of precision actually work in our favor, because we're probably just manipulating the whole lot of them, the whole thing, because it's the same kind of voxel that we can do anyway. So even if we use the real life situation, let's say in virtual reality, we can actually put a cat into the scanner together with the subject. But if you put a spider in the virtual reality, I think it would work very similarly than a still picture, because of the
if you test it, do you think virtual reality is going to work better than images, or will it just be kind of similar?
I would predict that it would be kind of similar, but we are partly also because virtual reality in the scanner is limited, because you can't really move your head. So it's really just kind of 3D goggle realistic movies rather than real virtual reality. So I would predict that they will be similar because of the bluntness of fMRI voxel responses. But we don't know. If VR is better, we'll definitely go for it. And that's why we're trying to.
Okay, thank you.
Thank you.
Last minute questions. I'm surprised we are quite late on a Friday, and still there are 30 people listening to us chatting. So no last minute question. I'll ask a question for the future, as you should as host. The pain, man, the pain. This is exciting, right? Pain is brutal. I mean, it's one of the biggest problems in general at the clinical level. Much more than phobias and other PTSD, etc. Even if PTSD has good press, it's like vegetative state, right? It's really small part of the population. Huh?
I would be a bit more careful. PTSD is also really traumatizing, literally.
Oh no, it's horrible, but it's much smaller as a societal economical problem than pain.
That's right.
That's what I mean. And
arguably, depression is also very big, and depression and anxiety goes hand in hand. But I agree with you. Pain is, and also as a consciousness researcher, that is the #1 evil qualia that we need to deal with. So I like to address that.
I just had a shiver through my my spine when you said qualia, but never mind, because you didn't care about the qualia. You just said, I can't see it in the brain, regardless of whether that's appraisal or true pain. And this separation has been there for years.
And I said it ungrammatically anyway. It's the number one quale. Yeah, it should not be plural. I butchered it. So I take it back. Sorry, go ahead.
So where does this goes, the pain? If you can really do it in EEG, then the possibilities are way better, exactly for what you just said, right? You're in the scanner, you're there, you can't do much, etc. But with the EEG now, I just co-published with friends a study on cycling, while you were in the EEG, and surprisingly, the signal, it's awesome once you clean up stuff. So where do you envision this? Just now we had 6 beers, and you can tell me, we're gonna do this with pain and blah. Just go for it.
I'm quite keen on that, because the whole direction is basically, I feel we have to move away from these esoteric, sometimes too personal debates, and really want to make a different, almost like societal impact, not just like to publish in some clinical journals and claim that we can get some NIH or whatever funding. I mean, if you really want to make a societal impact, the cost of fMRI is an issue, and it doesn't look like it would go down in time. And with EEG, in fact, EEG itself is a lot cheaper, and with the new technology, those dry EEG, both the hardware and the software have improved so much. In fact, it could become something you can just do it at home. And the further thing, I think I think it possibly a good insight there. We don't know whether it works yet, but even just an idea, I've been talking about it so much because I'm quite excited. The postcard idea, I think, might be onto something. So every time you're in pain, you have to wear the EEG thing and to do it. It may not work as well. Whereas if you do this every day as a kind of top up of the association, so you just carry that postcard, and every time you just look at it, could be like a kanji character, could be a Chinese character, you just look at it and just focus on your mind. It already brings out that anti-pain pattern you have associated every morning with that word. I think that could be, if it works, it will be amazing. But we are excited, but we don't know.
The word that came to mind when you said it, it was the talisman, which has been used shamanistically, for millennia. Hold into this. This is what, and this is top down, right?
Yeah.
Top down. What you're saying is not really purely top down.
Great.
So that's how I think the idea of not a top down talisman is really exciting. But
awesome. to the talisman.
Yeah, exactly. I'm good at titles. We know that science is difficult, but I'm good at titles. So, right. Any final words, Hakwan, or shall I close this in a sort of virtual manner?
Yeah.
Right, guys. Stay home.
Long live your prefrontal cortex.
Yeah. Thank you very much. It was really good. Stay safe, everyone. Stay at home if you can. Aha.